And I will share screen. Okay, so this lecture is about regeneration, repair, and homeostasis. And what's the lecture plan? Well, we're going to first find out what regeneration is. We're going to look at types of regeneration, and then we're going to look at regeneration in a range of different organisms, ranging from the Cnidarian Hydra through to flatworms. We're looking at regeneration in newts, specifically limb regeneration. Then we'll look at heart regeneration in zebrafish, and finally look at the limited regeneration that mammals can do and why that's the case. So for tissues to be maintained in a body, put simply, the cells produced have to equal the cells lost. If you lose more cells than you produce, then you start reducing the size of your tissues and their efficiency and effectiveness. If you overproduce cells, well, then the likelihood is that you're forming a tumour of some kind. So typically during normal homeostasis, cells produced equals cells lost. So what is regeneration? In biology, regeneration is the process of renewal, restoration and growth that makes genomes, cells, organisms and ecosystems resilient to natural fluctuations or events that cause disturbance or damage. Every species is capable of regeneration, from bacteria to humans. Regeneration can either be complete, where the new tissue is the same as the lost tissue, or incomplete, where after the necrotic tissue, so the injured tissue, comes some kind of fibrosis or scar. So every organism can regenerate, and this bar here determines the sort of degree of regeneration that can occur. So a Hydra can regenerate the entire individual from a small fragment. A flatworm can produce large portions of its body plan if they're lost. Amphibians and fish can regenerate limbs, tails, fins and various other components, including the central nervous system and heart. And then we come to mammals, and deer are able to regenerate their antlers every year, and mice and men can regenerate to a limited extent some organs. So it could be hard to visualize what's going on in other animals compared to mammals. And one way we can think about this is looking at superheroes who have regenerative abilities and comparing them to their counterparts in the animal kingdom. And I'm going to use for my examples Wolverine and Deadpool. So this is Wolverine and this is Deadpool. And in superhero regeneration, such as these two have, Wolverine has a healing factor, and he can regenerate lost limbs and he can heal scarlessly. Deadpool is a whole different situation. He can totally regenerate his body after being cut in two, although very weirdly he can't regenerate his terrible scars, so that seems like a weird biological oversight. So where would Wolverine and Deadpool sit on our range of animal regeneration? So Deadpool is very like a flatworm. It can be cut into pieces and regenerate from those individual pieces. Wolverine's a lot more like an amphibian or fish, and he's able to regenerate limbs and whole organs scarlessly. So it's unlikely that medicine can make us like Deadpool, but how could we be more like Wolverine? And understanding how these animals regenerate may give us clues to how mammals like us could regenerate scarlessly. So typically, the potential of stem cells that reside in the adult organism determine the regenerative capabilities. So Hydra have totipotent stem cells in the body, which we'll talk about a bit later. In this middle scale, there are pluripotent stem cells that can contribute to the repair of multiple organisms. And in mammals, where repair is limited, this is because the stem cells that are available for that repair to occur are they themselves quite limited, only multipotent, and so can only give rise to very few tissues. So different forms of regeneration can occur, and we'll go through these step by step. So first of all, we have a stem cell mediated regeneration, where in response to damage, stem cells divide and their progeny differentiate either into a range of different tissues if the stem cell is more pluripotent or totipotent, or if it's more unipotent or multipotent, the stem cell may give rise to a single differentiated tissue. We can also de-differentiate cells. So stem cells typically produce differentiated progeny. In a de-differentiation process, these differentiated cells become more stem cell-like, and from this stem cell-like state they can produce either more of the same differentiated cell or different kinds of differentiated cells. And we've seen these processes of de-differentiation before in, for example, the specification of neural crest cells. So this process also occurs during some repair processes in some organisms. We can get transdifferentiation, where typically a stem cell divides and forms progenitor cells, and then these progenitor cells divide and form mature cells. Transdifferentiation is the changing of one mature cell type to another mature cell type without the going back to a progenitor cell differentiation state. And again, this was another one of these possible mechanisms for neural crest potency that we looked at a few lectures ago. Do the neural crest cells transdifferentiate, or do they always have that potential to be like a blastula cell? We also have compensatory regeneration, where differentiated cells can actually reinitiate proliferation, and this allows for repair of the tissue without reliance on a stem cell or reliance on a transdifferentiation process. So all multicellular organisms use a number of these different processes at different times and places. So let's look at Hydra. We call it Hydra, its Latin name is Hydra. Offspring in Hydra are produced by budding off the adult. So we have it's an asexual reproduction. We get budding of the Hydra offspring off the adult, and it looks like a fully formed small version of the larger Hydra. So these are part of the phylum Cnidaria, and these have rotational symmetry. They're not bilateral. They only have an anterior-posterior axis, and you can see this here. You have a tentacle head end and a foot end, and there's only 2 germ layers in this Cnidaria, and you can see this here in cross sections. Here is a cross section across the body, and here is a cross section along the body, and you can see here an ectodermal layer, and underlying that ectodermal layer we have endoderm. So Hydra only has one axis, and it only has 2 germ layers. And this makes the processes of regeneration somewhat easier. And actually, a new Hydra is produced every 2 months. This is because the cells are very dynamic in the whole Hydra. They're a bit like the mammalian villus, where, remember, with the villus, the cells move from the crypt and then get shed at the top of the villus. In Hydra, cells continually move and are either shed from the tentacle or the foot. And this led Trembley, a biologist back in 1741, to state the story of the phoenix who is reborn from his own ashes, fabulous as it is, offers nothing more marvellous as a Hydra. When cut into as many as 40 pieces, there are reborn as many complete animals similar to the first. So, not using 18th century English, what he's saying is you can cut a Hydra into lots of pieces, and each of those pieces will produce a complete Hydra. So how does Hydra do this? Hydra has stem cells everywhere, all over the place. And if you look at a cross section of a Hydra, you will see these cells sitting in the ectoderm and also in the endoderm between them. These are called interstitial cells, and these are spread throughout the body of the Hydra. And these are the pluripotent stem cells from which a Hydra can repair itself. So if you cut it into little pieces, it doesn't matter how small a piece of Hydra you cut, there will always be stem cells present to help regenerate the whole organism. But how is there, in this case, control of differentiation to make a complete Hydra and not a Hydra cell containing lump like a teratoma? Well, it turns out that the very act of cutting a Hydra induces head-specific gene expression. So this is showing the expression of the gene Wnt3, and we can see here Wnt3 is expressed in the head region typically. If we take a cut through the Hydra, then one of the ends that's cut that becomes the head, and it's the area that's nearest the head, activates Wnt3 expression. Wnt3 is induced by the cut and it initiates head regeneration. So let's look at regeneration in our next most able to regenerate organism, the flatworm. And so what we can see here is we can take a normal flatworm, we can cut it into 3 pieces, and these 3 pieces will regenerate into 3 perfectly formed flatworms, irrespective of the piece that's cut. With these 3 large pieces, the flatworm can completely regenerate. What is interesting here is that the removed head end always regenerates the head, and the removed tail end always regenerates the tail. So it's fairly obvious here and here we have a tail end and a head end, and so the flatworm regenerates backwards and forwards as required. But when you cut the middle section, the head end, the end that was always a head, always regenerates to the head, and the end that was the tail always regenerates to form the tail. So is this the first evidence of a true anterior-posterior gradient? Back in 1905, Morgan and Child postulated that there was a factor in a gradient from the head end to the tail end of the organism, and after cutting, it's this gradient that specifies the head and rear part of the worm. So they thought that when you cut an embryo, what happens is typically a flatworm, Typically, the head end has the highest concentration of whatever this gradient is, and the tail end has the lowest end. So if you cut a chunk out of the middle of the embryo, the flatworm, then one end will have a slightly higher concentration of this factor than the other. The higher end will become the head, and the lower end will become the tail. Now, flatworms aren't quite as good as Hydra, and if you cut small parts of the flatworm, stranger things can happen. So if you cut only the small part of the head, we get different kinds of regeneration attempts to make the trunk and tail end. If you cut the middle part out, a very thin part of the middle part, then we get abortive attempts to regenerate the head and tail. And then if we take a part of the tail, that gets very confusing, and a head end is never normally produced, and a tail end isn't produced either. So smaller parts can't fully regenerate. Why could this be, based on this concept of a gradient? Well, it could be that when they're cut even smaller, there's no clear gradient of this factor, so the regeneration process gets confused and can't tell what should be head and what should be tail, and so the result of this is an incomplete regeneration. So we understand that that it requires gradients, the flatworm, to be able to repair effectively, but we also know that it needs some kind of cell that can do this. And there have been two models to determine what might be happening in the flatworm. And one could be that there is a pluripotent cell that can repair everything. It can be all cell types. The other option is that there are specialized cells that can only give rise to a certain number of different cell types, and these together contribute to the regeneration of the flatworm. So how can we experimentally determine this? Well, what we can do is we can try and kill the majority of the cells in the flatworm when it's repairing, and this was done in one of two ways. You could either irradiate with 1750 rats, which killed the vast majority of the cells in the flatworm, and the flatworm is able to completely regenerate all tissues. So then the experimenters took it one further. They could lethally irradiate, so kill all the cells in a lethally irradiated worm, and then take a single cell, a single neoblast from another worm, and they could show that that single worm, a single cell, could regenerate the entire body plan of the worm. And furthermore, if you cut this worm that had been repopulated with this single neoblast, cut it, then this was able itself to give rise to repair. So we have a cell that is able to proliferate and also give rise to all the progeny present in the flatworm. So a single cell can rescue the flatworm. Therefore, this stem cell must be naive. It must be unspecified or ES cell-like to be able to regenerate the complete body. So neoblasts are weird in that they are adult cells, they're somatic cells, and they are also pluripotent. They're a little bit weird, so let's have a look at some of the characteristics of planaria neoblasts. So interestingly, in flatworms, they are the only proliferating cell type. So they can give rise to every tissue. and all the differentiated tissues can't regenerate except through this neoblast pathway. These cells are pluripotent. They have indefinite self-renewal, so they're exactly like a typical stem cell. However, they are adult somatic cells, and they are mesenchymal cells. They have a large nucleus compared to the cytoplasm, which is a classic characteristic of stem cells, and surprisingly, compared to typical stem cell quantities in other organisms, they comprise 20% of all adult cells. And so we can see here that what happens is when we cut and wound and amputate, we get these neoblasts proliferating at the area that needs regeneration, and they're then able at that wound site to repair and regenerate all the missing tissues. This region of active repair where stem cell mediated regeneration occurs is called a blastema, and we'll look at that a bit later in other organisms. So head versus tail, how does a flatworm know where its head is compared to its tail? And briefly, it's the reverse of Hydra. So Wnt signaling is strongest in the posterior of the worm, and that high Wnt signaling determines where the tail is going to regenerate versus the head. So it's going to determine the head blastema versus a tail blastema. So let's look at regenerating limbs in amphibians. We've gone one step up in our ability or lack of ability to regenerate. So a newt can regenerate an entire limb within 7 to 10 weeks. How does it do this? Well, when you amputate the limb, you can see a bud forms, and then after 3 to 6 weeks, this bud grows and extends, and after 6 to 9 weeks, we get a fully formed limb that's functionally the same as the original limb. So this is very normal for amphibians, and they do this quite frequently. Like a flatworm, a newt makes a blastema. So again, we have this amputation, and then this wound epidermis becomes the region of stem cell mediated repair, the blastema, and then that blastema remodels the developing hand, essentially recapitulating those parts of limb development that are required, that have been lost in the amputation. So here we go. We have an amputation which leaves a wound where we have exposed epidermis, dermis and other tissues. This is quickly covered with a wound epithelium. Then dedifferentiation occurs where we get a formation of a blastema and what's called an apical epidermal cap. This then allows proliferation to happen in the blastema, and then eventually the blastema starts regenerating the lost bones until finally we have a fully intact regenerated hand. So just like a flatworm, a newt makes a blastema to repair its tissues, and also just like a flatworm, the newt knows, or the developing blastema knows what needs to be produced when. So let's have a look at the histology of the blastema, and here's our adult tissue structure. We have epidermis here, and we have our bone here. This is the site of the amputation. We have dedifferentiated tissue and changes to the blastema region. So we have a region of dedifferentiation where the cut occurred. Then we have this blastema region where active remodeling will be occurring. And then we have our apical ectodermal cap, which is the organising centre of all of this process. And this can be likened to the histology of the developing limb, where we have our adult tissue structure, we have a progress zone, which is the area that's actively being formed into the correct part of the limb, and that apical ectodermal ridge, remember, that secretes those fibroblast growth factors and antagonizes retinoic acid, and between them they determine what happens in this progress zone in the developing limb, or the blastema in the regenerating limb. So let's go one step further to so humans, when they have a heart attack, a myocardial infarction, they get damage to part of their heart, and then this heart tissue doesn't quite repair the same way. There's fibrosis and remodeling, and this injures the heart permanently. So every time you have a heart attack, your heart gets worse because you have more damaged tissue. A zebrafish, if it has or is given a cardiac infarction, so it has an induced heart attack, this causes damage in a region of its tissue, but it can completely regenerate this back to a fully functioning normal heart. What's interesting is that up until late embryogenesis, fetal mice can also perform the same trick as zebrafish. So if you induce cardiac damage, we see the damage here, but the developing mammal is able to regenerate it scarlessly. So why is it that the human has to undergo all this scarring and the embryo in zebrafish doesn't? And so to look at why or how zebrafish regenerates its heart, clonal analysis was performed with a way of Cre recombination which allowed different colour combinations to be expressed under the control of a constitutive promoter, in this case beta-actin, and we get a range of different colours. So in effect what we have is an organism where patches of cells adjacent to each other will be coloured the same way. And so using these patches of cells, we can track where these cells move over time and how they move in response to injury. And so rainbow hearts and clonal analysis allowed us to trace the injury and response to the injury of an adult zebra heart. So we can see here's our heart muscle cells, and in a normal uninjured heart, and we can see various different patches of cells corresponding to different lineages that were caused during that limited Cre recombination. And here we have the damage caused to the heart at this dotted line, and then 14 days post amputation of this region, we get movement of some of the cell populations into this damaged region to regenerate the heart. And then later on we get re-establishment of normal heart structure as new tissues, new cells move in and regenerate the heart. So we have multiple cardiomyocyte clones in the repair, and we can see that beyond the green and blue we can see some red, there's yellow cells here, and there's light blue cells here. Each of these colours represents a different clone of cardiomyocytes. So various different populations contribute together to the scarless repair of the adult zebrafish heart. So the big question is, why can't human hearts do this? Because we know that mammals can do this. Regeneration in mammals is better in embryos, and they can repair hearts and they can scar-free heal, but also they are in the womb, so they're protected, mostly protected from the harsh environment. And this suggests a reason why embryos can repair better than adult mammals. And this is because being a terrestrial animal is a big problem. Remember when we looked at how long it takes to regenerate a limb in the newt, and remember newts are quite small, it takes 9 weeks to regenerate a limb from an amputation. Now, a human, if you scaled it up, would take far longer than 9 weeks, but if a human had to take 9 weeks to restore a limb, it would be highly likely that it would die of infection and water loss, because while the limb is being formed, that during regeneration in the newt, that skin is not a good barrier to the external environment. So the priority in mammals to protect themselves from the outside environment and potential infection and damage caused by infection, the priority is to close the wound as quickly as possible and to not regenerate in the same way. And this requires rapid interaction of various cell types. So we have epithelial cells that are rapidly closing the wound. We have immune cells and myofibroblasts, which tie together the dermis part of the tissue and clear out any infection. And this requires the use of matrix metalloproteinases to remodel this dermis. And so together, this quick remodeling means that it doesn't regenerate exactly the same as it was before the damage. And it's this imperfect regeneration because of the rapidity, the speed with which you have to repair these holes in mammals, that we get scarring. So we get misdeposition of collagen, and we get differentiated myofibroblasts pulling the wound together, and so this can lead to pathological changes or scars. Humans can, however, regenerate their liver in a process called compensatory regeneration. So they can have cells in the liver that are differentiated, and these cells divide to repair the damaged liver. And this is obviously important for liver homeostasis, and what was found was that the hepatocytes near the vein have stem cell-like properties. So even though they're all hepatocytes, these pericentral hepatocytes respond to Wnt signals and have stem cell-like properties. These allow for turnover of these cells, and they can divide and repopulate the hepatocyte population that contains the liver. However, in instances like cirrhosis of the liver, the even damage to these pericentral hepatocytes occurs, and in these chronic liver injury situations, Wnt signaling cannot activate this stem cell-like property of these cells. They become incompletely able to repair, and therefore parts of the liver are permanently damaged. So you need a relatively healthy liver and not much damage to be able to repair it using these pericentral hepatocytes. Liver regeneration reduces scarring as much as possible. So if we have regeneration in the normal liver, we get efficient hepatocyte division because we're not getting a lot of entry of immune cells such as macrophages into their repairing tissue. So we get very little scarring, and the repair of the tissue is very much the same as the normal liver. However, if you're chronically damaging your liver, either through hepatitis or chronic use of excess alcohol, then we get many more activated macrophages entering the region that needs to be repaired. We get much more activation of myofibroblasts. This leads to excess scarring and necrosis and apoptosis, removal of these dead cells, and less space and ability for these hepatocytes to fully divide and regenerate the chronically damaged liver. So again, if you damage the liver too much, then the immune response and the healing response is so extreme that these cells cannot divide to repopulate the liver. So that's the end of this lecture. What did we go through here? We looked at what regeneration is. We looked at types of regeneration that are typically stem cell mediated, and this kind of hierarchy of stem cell regeneration where more potent stem cells are able to allow the organism to undergo more extreme forms of regeneration. And we saw that extreme regeneration in Hydra, where you can cut it into a tiny piece and you can still establish a Wnt gradient, which then activates those interstitial cells to regenerate the organism entirely. We then looked at flatworms and how you can cut portions of them, and if there is a sufficient gradient of Wnt signaling, then a blastema is formed, and that blastema can regenerate and repopulate the complete organism body plan. We showed that this required pluripotent stem cells called neoblasts that can regenerate into all tissue types in the flatworm. We then looked at limb regeneration in newts and the concept of amputation forming a blastema, and this recapitulation, this redoing of the limb development is seen in limb regeneration. We saw that zebrafish can scarlessly heal its heart due to the contribution of various populations of cardiomyocytes. Mammals can't repair scarlessly because they need to rapidly repair damage, and this is why we get scars in the skin. Liver can repair scarlessly, but only if there's not too much damage to begin with. So if we look at what's possible, I think maybe humans could be Wolverine in the future, but they certainly couldn't be Deadpool. So with that, I think I will stop there. We have an office hour at the normal time, and then after that office hour we have a workshop. So I will see you at both of those. I will see you later. Okay, see you. Bye.